Again, good morning, everyone, good afternoon, or good night, wherever you are. Today, we're having a talk by Helena Kwarteng from the State University of New York in Binghamton in the US. And she's going to talk about quantifying diachroneity of calcareous nannofossil datums across the Western Pacific Ocean. So the format of today's seminar, if you're new here, we'll do a quick welcome and announcements right now. Then we'll have Helena's talk for around 30 minutes, followed by a moderated Q&A where you can send your questions. You can either send them directly in the chat. If you just want to ask them yourself, you can also send them to me first if you want to stay anonymous. And after that, Helena has agreed to stay for tea time. So if you want to ask some more detailed technical questions, you can ask them there. A little bit of housekeeping before we start with the talk. So PaleoPerks values the participation of all folks interested in the paleosciences. Please remember to abide by our code of conduct during today's seminar. Also, please keep yourself muted for the duration of the talk out of respect to the speaker. As I said, you can ask questions by either sending them to me, we have no questions host today, or just by sending them in the chat. And if you want to ask them by voice, you can use the raise hand function and I will unmute you. My name is Nina, by the way, and I'm today's host, as you can see. Then we also have closed captions built into Zoom, and you can use the CC button to either show them or hide them. And then finally, PaleoPerks only works because we have so many cool early career researchers that are willing to share their work. And we're always looking for more. So if you know someone or if you are an early career researcher that wants to present, we also accept self-nominations. Please go to this link. I will also drop these in the chat later. And then finally, we would also appreciate if you fill out the weekly feedback form. This is completely anonymous and optional, but it gives us some information on who is watching from where and if you have any feedback. I will also drop this in the chat later. Then a bit about the speaker today, Helena Kwarteng. Helena did her bachelor's at the University of Ghana in Ghana. She then moved to the US to do her master's at the University of North Carolina at Wilmington. And she's currently a PhD candidate at Earth Sciences at the State University of New York in Binghamton. And with that, I would like to invite you, Helena, to share your screen and start your talk. Thank you.
Thank you so much for this wonderful opportunity. Okay. Yeah, so hello everyone. My name is Helena Kwarteng, and as I've already been introduced, I'm a PhD candidate with a geology department at Binghamton University. And I'm very excited to talk to you about my work titled Quantifying Diachroneity of Calcareous Nannofossil Datums across the Western Pacific. Before we get into the technicalities of this work, I would like to give you a quick refresher about what biostratigraphy is and also why the fossils that I'm interested in. So let's start with biostratigraphy. So biostratigraphy is a dating tool that is used to assign relative ages to geologic strata based on the first and last occurrences of fossils. It has mostly been used throughout the Phanerozoic record. And the image that you see on the slide is a simplified model that shows how these correlations work. So you realize that the occurrences of the fossil are being used to correlate the different strata that you see in the model. So for the late Mesozoic to the Cenozoic, the fossil that is mostly being used is calcareous microfossils. And for this, we have plantiforams and we also have calcareous nanofossils. So getting into the why of these microfossils being used as dating tools, it's because they are broadly distributed geographically. They are also very abundant in marine sediments. They evolve very quickly, which is also another tool that really another factor that helps with biostratigraphy. And also they are in codes easily identifiable because I know the easy here is a little bit tricky, yeah. And from the movies that you are seeing, we have the first two showing foraminifera. With the second one, you could see how the foram strap, some food that it wants to get. Then with the third movie that you see on the right, you can also see a coccolithosphere attaching plates to itself to get the full coccolithophore formed. So this is what the life of these microfossils look like. But then what we usually investigate is the fossilized forms. And these are what the fossilized forms also look like. So on the left, you have the Calcareous nanofossils that are being, so these were being observed under a polarized light microscope. Then on the right-hand side, you also have plantic foraminifera, also observed under the light microscope. So now that we've been able to go through or refresh our memories on some of these basics, we'll start getting into the technicalities of my work. So before we get into what diachroneity is, one thing that I want you to note is biostratigraphy relies heavily on the assumption that primary datums are synchronous across the globe. And what that means is they believe that these markers do occur at the same time across the globe, and that is what makes it possible to be able to correlate one to another and being able to interpret that this strata here occurred at the same time that strata B also occurred. However, this is always not the case. There are cases where you can have some of the datums appearing earlier and you can also have some of them appearing later. So the model, again, this new model shows how some of these offsets can happen, which can lead to misinterpretations of when stratas were being deposited. So this idea of diachronacy was first brought up by Dowsett and Way in the 1990s. However, they never got to the point of doing a quantitative analysis to know how much offsets we have in these datums. It wasn't until 2022 where Lam et al. Decided to quantify diachronacy among biostratigraphy datums. And they did this mainly for plantic foraminifera. So with the work that Lam et al. Did, they realized that there have been cases where they can have plantic forams, having offsets of up to about 0.8 million years. And that is very wild because imagine you dating a sediment and or a particular strata that you're interested in and you're off by, hundreds of thousands of years or yeah, millions of years. That is crazy because depending on your work or what you're working on, it can drastically affect the results that you get, which wouldn't be right. So these were some of the findings Lam et al. Had, and they did their study across the entire Western Pacific. So they found out that plantic forams are diachronous within regions, between regions, and also relative to the subtropical zonation scheme. So the subtropical zonation scheme is just the global scheme that provides the ages of where we expect the ages that we expect datums to occur. So they realized that the ages that they were finding at some of the Western Pacific sites did not match what we have in the tropical, subtropical zonation scheme. And within regions, maybe in a particular region like the Northwest Pacific or the Southwest Pacific, they realized that some of the sites were still exhibiting diachronacy, even though they were within the same region. Then, of course, between regions too, they were also seeing some diachronous streams. So among the diachronous diachronous patterns that they observed because they investigated this across the entire Western Pacific. They were able to observe some patterns, and the patterns that they found out include stepwise migration and gradual migration. So the stepwise is when you have the datums kind of moving in the form of stairs, then the gradual is also, just as gradual as they evolve through time. So this was what drove my interest. If you remember, as I talked earlier on, two of the microfossil groups that are mainly used for biostratigraphy from the late Mesozoic to recent times is Calcareous nanofossils and plantic foraminifera. So if we've gotten to a point where we are seeing that plantic forams are very diachronous and it can really affect the works that we do, then how about the other group? So this is what is driving my research interest. I want to do a comparative study that will investigate the other microfossil groups. So basically what I want to do is doing the same thing Lam et al. Did in 2022, but then this time around with calcareous nanofossil. And for the purpose of this talk, I will be focusing on the Northwest Pacific region. So these are some questions that I'm asking. So my research question one, are there offsets in calcareous nanofossils relative to the tropical subtropical zonation scheme? Then I'm also interested in knowing if the offsets in calcareous nanofossils are comparable to those found in plantic foraminifera. I'm also very interested in knowing whether the datums that are being observed, the calcareous nanofossil datums that are being observed, behave synchronously or asynchronously across different sites within the Northwest Pacific region. So I decided to, as every researcher will do, once you have the questions that you want to ask, you also think about what you're going to do to be able to answer those questions. So for my methodology, I ended up compiling all the calcareous nanofossil datums for three sites that we have in the Northwest Pacific. So for the holes, you see 12070A, you also see 1208A, and you also see 1209A. So these are the three holes that I'm investigating in the Northwest Pacific region. And it is important to note that the biostratigraphy schemes that I found in this region, they were all shipboard data except the 1208A, which was refined by Bound 2005 after the shipboard data. So I compiled the datums in the calcareous nanofossil datums in these resources, and I recalibrated them using published magnetostratigraphy age models that have been tied to GTS 2020. So once I was done with that, I compared the datum ages to the subtropical tropical zonation scheme, and I also compared the offsets among the sites in this region. Then the other thing that I'm also interested in is knowing how these offsets compare to the other microfossil group, which is plantic foraminifera. So that's another thing that I also investigated. Then it is also important to note some of the challenges that I had and some of the standardizations that I did to be able to arrive, to be able to proceed with my analysis. So I wanted to investigate the neogen and quaternary. However, sometimes the magnetostratigraphy at some sites are quite patchy. Sometimes you don't get all the datums that you want occurring at all the sites. And also from the perspective of taxonomy, sometimes different terminology and different concepts are being used in different resources. For instance, if you can have common occurrence of a particular datum, but then that is being reported as the peak in a different resource. And also there are cases where you can have first common occurrence, but then you see another resource also reporting first occurrence. And it is important to note that these are not the same. So the table shows some of the examples that saw that I had to get rid of. For instance, as you can see in the first occurrence of the Scoesta dragi here, but then in the GTS 2020, this was reported as the Scoesta dragi subspecies. So in that case, you can't even use the two because they mean two different things. But then with stuff like Amorolitos species, usually um, the occurrence of all the Amorolitos genus is approximately the same. So sometimes even though some of the resources don't refine it to the species level, it's still possible to work around with stuff like this. So after doing all these processing, these are some of the results that I got, and my subsequent slides will be walking you through some of the findings that I have. So what we are seeing on the slide here is for 1207A. So these are offsets that I observed in the Calcareous Nannofossil Datums for 1207A. And I investigated 14 datums for this particular hole. So as you can see, the middle part, we see that the species here are synchronous. So you don't see any offset. But then when you check the bars that are above zero and below zero, you realize that the above zero means the datums occurred much more later compared to the subtropical zonation scheme. And the bars below, that's the blue ones, also means that they occurred much more earlier compared to the subtropical zonation scheme. So we see some datums being synchronous here, and we see some appearing much more later, and we see a couple of them appearing much more earlier. And we'll be getting into some of the details of some of these species in the subsequent slides. Now for 1208A, also not too different findings. We see that some of the datums, for here I investigated 19 datums. We see that some of them are diachronous. Most of them are diachronous, even though the magnitude of the offset here is relatively lower compared to what we saw earlier on. But then we see that some of the datums again appeared much more later and others appeared much more earlier. And now when we also look at 1209A, for 1209A, I was only able to get 6 datums from the shipboard data, but then we were still able to make some investigations here. From here to we realize that with the exception of these two that are nearly synchronous, the remaining ones are highly diachronous. So, and we realize that for the diachronous datums, they all appear later, unlike the first two that we saw, where we could see that some were appearing very early and some were appearing very late. So, having all these findings from these individual sites, I decided to bring everything together to have an idea about some common datums that do occur across the 3 sites, or at least 2 of them. And we realized that a few of them do appear at both sites. For instance, for the 3 sites, we have Lacunosa, Amolito species, and we also have brewery appearing. And for a couple of them, we only have them appearing at 2 of the sites, then the remaining ones just at 1 site. But then the point is, even with the places where they appear at 2 or more sites, we could see that there are times where they are synchronous, as we can see in the last occurrence of the brewery, but then there are also times where they are very diachronous, as we can see in the last occurrences of D. hamartis. You see that 1207, the offset is very small, but then when you compare the 1209A of the same datum, we realize that the offset is very huge. So I also decided to investigate the pattern of this in these diachronaty in the three sites where possible. So it is important to note that the sites that I am focusing to work on, this is, oh, sorry. It's important to note that this is a transitional zone where we have subtropical water being brought northward by the Kuroshio currents, and this meets the colder water that is also being brought southwards by the Oyashio currents, creating distinct water masses in this region, which could cause species to appear later or earlier. So this is a very good region to investigate diachronaty. So I realized that, so from the plots that we have here, you realize that for the Amolito species, the species occurred, the last occurrence of the species exited much more earlier in the subpolar hole or region, but then it disappeared much more later in the subtropical region. And for the transitional region, which is the 1208A, we see that it's nearly synchronous. So what this means is the Amaryllitos species has been shown to be a warm, has been shown to be associated with warm environments. So what that means is in the case where we are seeing it appearing, disappearing much more later in 1209A, which is relatively warm as compared to the other two hosts, then it means the environmental conditions in this region was much more favorable for this particular species, and that is why it ended up disappearing much more later compared to the subpolar region, which is further north 1207A. I also extended this to the other species where we have them occurring in two or three sites, but then for the three sites, I realized that beside the Amarylito species, the other ones were nearly synchronous, so it's not possible to investigate these patterns. So these are the ones that do appear at two sites. And for that, we also realized that with the last occurrence of the Scoasta hamatus and the Rectinosphaera pseudo-umbilicus, we realized that they disappeared much more later, again in the subtropical, tropical, compared to the subpolar. So this is also giving us a very good clue about taxa that are being warm, always disappearing much more later in the subtropical zone, which is very different from subpolar, which is very cold, and of course they disappear very early there because the environmental conditions is not too favorable. But then we also had some interesting findings for the first occurrences at these for different species that are also warm associated, and we were expecting the first occurrences in the subtropical to be much more earlier than the subpolar, since the subtropical environment will be much more warmer and more convenient for them compared to the subpolar regions. But then we realized that the findings for the first occurrences of the Ceratolithos rugosus and the first occurrence of Amorolithos species are inconsistent with what we would typically expect. So this is possibly due to some mixing going on in these regions, or it could also be attributed to the fact that the biostratigraphy for some of these two sites, that's the 1207 and the 1209, are poor resolution because they've not been refined yet, or taxonomy inconsistencies between aspects. So once I found all these, or I had all these findings, I decided to make a summary of everything to see how things are behaving generally across the three hosts. So for that, I also realized that 1208 shows the most, like the tightest clustering around the GTS 20 or close to being synchronous. Yeah, compared to the GTS 20. Then that is being followed by the 1209, the 1207A. But then with the 1207A, which is in blue, we realize that some of the datums do occur much more earlier, some of them do disappear much more later. Then for 1209, we realize that there's a very wide spread here. Things are much more diachronous, and it is also important to note that the datums only appeared later in the 1209A. So after getting these findings, um, if you remember from what I said earlier on, inspired this research, being able to see how calcareous nanofossil will behave and compare those two forms. So with the calcareous nanofossils, these are some of the preliminary findings that we have. And I was also interested in bringing the two different microfossil groups together to see how they are behaving in terms of the magnitude of the offsets. So with a magnitude for this plot that we are seeing, the calcareous nanofossil is in the peach color and the red. Then the plantic foraminifera is in the purple and the blue. So you realize that the magnitudes of the offsets in the blues and the purples are much more larger than the magnitude of the offsets in the peach and the red. So this also kind of clue us into the fact that the plantic foraminifera exhibits much more diachronaity as compared to calcareous nanofossils. And I decided to make a summary box plot here that will help us visualize things better. So again, or in summary, you realize that the calcareous nanofossils do cluster tightly around the zero, which means there's close to synchronity there, but then we see a lot of widespread in the plantic foraminifera. So these are the findings that I have, and we the findings that we've gotten helps me answer some of the questions that I'm interested in answering. So for my research question one, which is, are there offsets in calcareous nanofossils relative to the tropical subtropical zonation scheme? Yes, we've been able to see a couple of offsets. Then the second one is, how are they comparable to the offsets found in plantic foraminifera? We realize that the offsets in calcareous nanofossils are much more less diachronous compared to that of forams. Then, do calcareous nanofossil datums behave synchronously or asynchronously, especially within the Northwest Pacific? So for that one, we also realize that some datums do show synchronity, but then others are also diachronous. Then some future work that I will be doing, I intend to extend this study investigation further southwards in the Western Pacific. And I'll also be refining the biozonation schemes of the Northwest Pacific sites and possibly the Southwest Pacific sites as well. And the third thing I'm also very interested in doing is again comparing the magnitude between forams and calcareous nanofossils for all the sites in the Western Pacific and also investigating the biogeographic patterns of the two groups. So in that case, comparing the patterns that I see with Lamb et al.'s own to see whether I'll also be getting those stepwise and gradual patterns or what I observe in calcareous nanofossils will be much more different. Yeah, and these are my references and acknowledgements. So that's all that I have for this talk. Thank you, and I'm open to any questions that you may have. And if you're watching this later on and you have any question, you can always reach out to me at my email, hquarten@binghamton.edu. Thank you.
Thank you very much for your talk, Helena. That was super interesting. Yeah, I think this is also very good timing to be working on stuff like this. Like as some of you may know, next week is or in 2 weeks, there's the Penrose meeting in the US
Oh,
concerning the future of timescale calibration. So this is exactly the type of work that we need to be doing for that.
yeah.
Again, if you have questions, please send them in the chat or use the raise hand function or send them to me. I have one that I will start with. So I found it very interesting that diachroneity seems to be lower in the nanos than in the forams. Do you have any gut feeling or hypothesis on why that is the case?
Yeah, so it's mm, currently what I have is because calcareous nanofossils are photosynthetic organisms compared to the forams that are heterotrophs. But I've not done any further investigation to, be able to confidently make these claims. But yeah, that's what I have. Yeah.
Yeah, that makes sense as a starting hypothesis.
Mhm.
See. Another question. Great talk. Could it be that part of the diachrony comes from preservation issues, either of the
Yeah.
nanofossils or the magnetostratigraphic signal that you use as a reference?
Um, yeah, so for the preservation, yes, that is a very big factor. Um, so it is also important. So this all also gets into the nitty-gritty details of taxonomy when people are doing biostratigraphy, making sure you make all the notes of whatever that you are observing. Because imagine some of these biostratigraphy schemes that we have out there, which I am compiling all these data from. Probably there were some preservation issues somewhere that wasn't reported, right? So where it's been reported that this datum occurred, if I don't dig further to know that, oh, there was a preservation issue here, then I wouldn't know and I would think that is actually the first or the last occurrence. But then that may not be the true first or last occurrence. So preservation is one big thing that, really have to be considered when making interpretations on with regards to some of these findings. Then for the magnetostratigraphy, that one, it's mostly depends on the resolution at which the work was done. So if it's very poor resolution, you don't even try to use the polarities at all because you'll be miscorrelating stuff. So there, as part of why I only presented on the Northwest Pacific region, I was actually doing some analysis for the southern sites, and I realized with some of them, the resolution for the magnetostratigraphy is very poor, or very patchy. So in that case, if you're not very careful with the way you use it, you end up getting results that are not truly representing what actually happened. So it's very possible for some of these methodological artifacts to affect some of our findings, and that's why we try to rule everything that could be a factor out before proceeding with the analysis that we have to make. Yeah.
Thank you. Very thorough answer. And that makes sense, of course. I have a follow-up question for the taxonomy. Just as a long-term project, do you think that to solve these diachronies, people need to redo a lot of the taxonomy for nanofossils, or is that not really the problem? I'm not a
Yeah.
nanofossil person. I don't know how old this taxonomy is in most cases.
Yeah, for both groups, there's a lot of taxonomic revisions that have to be made. And not like introducing new stuff, but making sure scientists or experts are calling species A species A. And you know that is very difficult. So there are a lot of working groups across both microfossil groups that are aiming to, help solve some of these problems or reduce, some of the issues. Yeah. And yeah, the other issue too with it, so I also forgot to mention this, but then one thing that I also want to add, which is not even related to just this question that I answered, this whole research also highlights how it's important to have other independent stratigraphy models because there have been other sites that I want to investigate, but then if you don't find if the magnetostratigraphy there is not good or is very patchy, and you don't have a different independent age model, then it's hard to make this investigation. Yeah, so
Yeah, I think there is a lot of, especially legacy cores that could benefit from some
yeah,
new work on it, probably.
yep.
Um, let's see. There is one more question posted in the chat now. Asking regarding the pattern of datum offsets, is temperature, so warm versus cold taxa, the main control, or is there also an effect of, for example, food availability? I
Yeah,
imagine
so
like oligotrophic versus eutrophic species.
yeah, all those things definitely factor into controlling the patterns. So with regards to productivity, temperature being favorable, especially when you have some of these warm taxa. In the case where you have a lot of productivity in a particular region and the temperature is also favorable for them, of course, they will end up being there for a very long time. So those ecological factors definitely also factor into we seeing some of these diachronous strains. But then these are all hypotheses. None of them have been investigated into full details to know that okay, this is actually the cause, or this contributes to diachronicity by this percentage or that percentage. So these are all hypotheses that are in the works that have to be done by all of us. Yeah.
Yeah, that makes sense. So a lot of work to be done, but you made a great start on it, I think. Um, I give a few more moments to see. Okay, there is one more question by Mohammed Javad Razomoi. I hope I pronounced it correctly. Asking, great talk. Do you think the less diachronicity can be because nanofossils spread faster or because they are smaller and can be carried easier by the currents, I think, compared to forams?
Yeah, so that is also a very good point. Um, I've not thought about yeah, that is also definitely a factor. being able to be carried around easily should definitely have some impact on, when they do appear in certain regions. Then in addition to that, when we get back to the preservation issues, because calcareous nanofossils are also much more tinier compared to plantic forams. In the case where you have dissolution in a particular interval, the calcareous nanofossils are much more resistant than the plantic forams in the sense that they are very, very tiny and they can easily get away with it. But plantic, sometimes it's hard. So that is a very good point. And there's definitely more than a couple of things controlling all these things. We just have to get to the point of being able to disentangle all of them.
Thank you, Helena, and thank you, Mohammed, for the question. I'll wait a few more seconds if there is any more questions. In the meantime, I want to again thank you, Helena, for your talk, for getting up very early. To the rest of the audience, it's like 5 a.m. for Helena now in the Eastern US, so great that she's here. Yeah, if there's no more questions, and then before we go into tea time, I will share some information about next week's talk. And again, thank you, Helena. And also thank you for the audience for joining us, of course. Yeah, so it would be great if you fill out the weekly feedback form that you can find in the chat. Next week, we have a seminar that's again at 15 UTC. So check your time zones, what time that is for you. And this will be by Nick Thurber from the University of California, Davis in the US. And he will be talking about vertebrate paleontology. So if you want to stick around a little bit for tea time, please stay. So tea time is just an informal conversation about the talk in a relaxed setting where you can ask maybe some off-topic questions or some more technical questions to Helena. I will turn off the recording and stop the recording, and everyone can just turn on their cameras if you want. But before that, because our speaker has been talking for a long time, we want to give them a quick break. So we'll have a 2-minute break now. And to those of you that want to stick around, see you then. To those of you that can't or don't have time, hopefully see you next week.